Hi, everybody. Good morning. Thank you so much for coming and for your interest in the bioprocessing seminar. This is really a seminar that was organized casually. The reason is that two of the speakers were my students, and then now they're all working for the same company, Esco Aster, and so we will have an overview of the projects as well as the applications of what they have learned during their PhD in the company. So the company itself, the CEO will be presenting in a little bit, and he's stuck in the traffic right now, so he will present second. And Geraldine Chiew, who was in my lab from 2015 to 2017, the second half of her PhD, will be presenting first. And the second presenter will be the CEO of the company, Esco Aster. He's a relatively young chap, and he actually was the one who pushed for this bioprocessing unit in Esco Aster. And the third speaker will be my other student who just graduated probably a couple of years ago. Yeah, he was the one who developed the bioprocessing activities in my lab, and then because of that, he got snatched by the company right when we were looking for a contract manufacturer for our products. So anyways, it's a pleasure really to have them over here, all three of them together. And without further ado, I'm gonna have Geraldine Chiew to give the first talk.
So thank you, Prof. Serene, for inviting me to this seminar. So it's my delight to share with you guys on stem cell therapy. Okay, yeah. So I'll mainly go through these four topics. Stem cells, what are they actually? And then how do we actually utilize them for therapy, stem cells therapy? So the clinical usage and the indication for that. And after that, I'll bring you through the industrial route for manufacturing stem cells. So mainly I will touch on mesenchymal stem cells. And I'll give you a whole overview of this production process. And then finally, what's the future of these stem cells? Insights into generation 2 of stem cells. So firstly, intro to stem cells. Stem cells, they are actually cells that are capable of renewing themselves and differentiating into multi-lineage cells. I'll just remove my mask. Yeah. So it is also the cell's capacity to see how they are able to differentiate, and it's affected by their potency. So over here, you can actually see that most of the time, stem cells actually comes from blastocytes. It's also the embryo. When a fertilized egg is we have these stem cells that's taken from this fertilized egg. From there, this fertilized egg, they are able to differentiate into like cardiac cells, your heart muscles, fat cells, red blood cells, chondrocytes, your bone cartilage, neurons, your brain cells, things like that. So they are able to differentiate into many types of cells. So the extent of which these stem cells are able to differentiate is actually their potency. So to be able to differentiate into many different types of cells, so that is totipotent, the very godlike cells. So they are actually from our very first fertilized egg, and then followed by pluripotent. So these are able to differentiate into all body cell types. This is actually most of our cells that we take from the embryo. And then after that, there's multipotent cells. They can differentiate into several cell types, and then oligopotent and then unipotent for their potency. So you should know that not all stem cells are equal here. So the cell potency is the cell's ability to differentiate into other cell types. So the more cell types that they can differentiate into, the greater its potency. So for example, totipotent cells, These are cells that can give rise to any of the 220 cell types found in an embryo. So they can also differentiate into the placenta. And then pluripotent stem cells, we'll also be touching on that later. This is to all cell types. They are mainly from the ESC. They are mainly from the embryonic stem cells. And then multipotent stem cells, we'll touch a lot on that. That is where the therapy focus on multipotent stem cells. Yeah. So as you see, pluripotent stem cells, they are actually quite godlike in the sense that they can differentiate into many cell types. And then most of them are actually from embryonic stem cells. They are derived from embryos that are 3 to 5 days old and then harvested during in vitro fertilization. But this also actually caused an issue, ethical issue in stem cells research. So you are destroying human embryos in order to get this embryonic stem cells. So it is actually unethical, and it caused political controversy during 1980s when this was ongoing. So how do we actually counteract this? So we are actually using iPSCs. We induce to form these pluripotent stem cells. So most of them are actually derived from adult stem cells. So for example, I can take proserine skin cells, and then I will reprogram it into my induced pluripotent stem cells. And thereafter, I can differentiate them into many cell types. Yeah. And another example, so multipotent stem cells, mesenchymal stem cells. So these are from the adult stem cells. So let's say, for example, from our bone marrow, from our umbilical cord, they have these stem cells found within these areas. So it is also commonly used in many cell-based therapy for now. Just a brief history of the stem cells. Yep. So if you can see, actually, from the very first part, when the stem cells first arrived, it's actually about 1900s, when people found that embryos, they have this tendency to differentiate into many cell types, but nothing was actually found until 1956. We had our very first successful bone marrow transplantation. And then followed by, in research-wise, 1963, there was this scientist, Frenenstein. He found that from the bone marrow, you could actually isolate stem cells that has these properties where they can form colonies, and they are able to have this cell renewing potential. At that point of time, he actually termed them osteogenic stem cells from bone. Thereafter, in 1973, we had our first unrelated bone marrow transplantation. It was very successful. Before that, 1981, ESC from mouse was first found out. And Martin Evans got this Nobel Prize for his work. And of course, one that was very famous for the stem cells research world was actually Dolly the clone sheep. So Dolly was founded in 1997, and there were a lot of popularity on Dolly. Dolly even went on to Time magazines and things like that, because people were wondering, is that really a clone sheep or it's just another type of sheep? Yeah. So anyway, Dolly died 5 years later, but then for that, we already see that cloning can be done. So next up, 1998, the human ESCs was isolated from embryos, and then that's where research was actually paused for quite a while because of all the controversy and things like that, until in 2012, Yamanaka and John Gordon, both of them found this iPSCs, and which we can actually reprogram from human cells. So both of them won this Nobel Prize in 2012, and then again, stem cells was very hot then thereafter. Oh, sorry, my timeline is a bit messed up. So, but yeah, ESC was also used for the spinal injury, first in men for this kind of treatment. And then of course, till now, presently, there's more than 100 clinical trials on this kind of stem cells therapy. Yeah. So I'll touch mainly on mesenchymal stem cells, MSCs. So where are they actually sourced from? So from bone marrow, that's the most common one. It's also related to our history just now. Frenenstein first found that stem cells came from bone marrow, but thereafter in 2000 and so forth, people found that it's also from fat. You can find it in your teeth, you can find it in the placenta, in the umbilical cord, blood, and so forth. So these are a few sources of these stem cells: adipose tissue, your fat, your bone marrow, dental pulp, and then umbilical cord. So I would say bone marrow was the very first, but then getting this cell source is mostly painful and there's a risk of infection. But then of course the year and the potential is dependent based on the donor's characteristic. If I get a 50 years old bone marrow stem cells donor, of course the self-renewal ability won't be that great. And then adipose stem cells, it's very easy to get nowadays because you just do a bit of liposuctions and then you can go on to get your stem cells from there. So, but of course, there's a bit of differentiation potential for this kind of source in the sense that they have inferior osteogenic and chondrogenic formation compared to the bone marrow stem cells. And then dental pulp is quite fairly common nowadays, especially from our teeth. But yeah, sometimes it's a bit difficult and invasive to try to get your teeth cells. Yeah. So the differentiation potential, most of them are highly used for bone forming cells or colony forming cells. Yeah. And then lastly, umbilical cord. These are actually trending now, umbilical cord, because from the pregnant women, most of them will want to keep it as a memory, the umbilical cord, and then at the same time you can use it for other purposes. So all this umbilical cord is easy to get. You get tons of cells from that one line of cord, and then at the same time you found that it's actually quite able to differentiate, and then you can get a lot of cells from here. And then last one that I want to actually touch on is actually hemopoietic stem cells. Sorry, I didn't spell this out. So they are oligopotent stem cells. They are one class lesser than umbilical cord mesenchymal stem cells, but they are also very good for treatment. So a lot of approved therapy nowadays, they are actually using umbilical cord blood. The blood source, it can be used for a lot of treatment, especially genetic diseases, where if your newborn, it has a bit of if your newborn has spinal disease or like leukemia, things like that, you can use the brother's umbilical cord blood to cure this disease. So this is already approved by FDA and very ongoing till now. Yeah. So stem cells therapy. Okay, for the cell therapy is actually the sorry, let me go back a bit. Yeah. So cell therapy is actually the administration of these live whole cells or the maturation of a specific cell population for the treatment of a disease. So it can actually be divided into allogenic or autologous. I'll go into that in a while. So the three types of stem cells that I actually pointed out just now, these are the most commonly found in industry. So embryonic stem cells, not in use anymore due to ethical reasons. Induced pluripotent stem cells is good, but for now it's still not suitable for therapy. I'll go into that later on too. It's currently trending. So most of what I'll be touching on will be on mesenchymal stem cells. It has this long history of research and there are clinical trials for it, and it's also used in therapy now. So how does these stem cells come about? Maybe I touched on it just now, but I'll just go through very quickly. So getting the cell source from their different areas and then harvest it. You actually expand in the culture, in the laboratory, and then you do see that it still have this kind of differentiation potential. You can use it straight away as a naive stem cells, or you can actually differentiate into other cell source. And then all of this can be used for a number of diseases. So for example, inflammatory disease, like on the knee repair, things like that. Yeah. So there's a possibility for autograft, allograft, and it has immunosuppressive effect. And at the same time, there's no ethical issues and things like that. Yeah. So autologous versus allogenic. This is the two main difference that you might want to know. Autologous is when I actually take your cells and then I grow it, do some changes to it, and I can put it back into your body. Autologous. For allogenic, I take it from a certain one person. I take it from Prof. Serene, and then after that, I will just put it into Rupali or whoever patients that requires it. Yeah. So allogenic over here. So how can MSCs be used? So I touched on it a bit just now, but mostly I would have to say that it's for regenerative purposes to replace damaged tissue. So leukemia, lymphocyte, blood, and immunological disease is very common. And then genetic disorder repair. An example I was giving just now was the hematopoietic stem cells. Yep. And then in anti-cancer and anti-infection, anti-inflammatory, these are some very common ones out there. So all in all, there's quite a lot of clinical trials happening, and this is one of the pie chart that shows how they are divided into for the diseases. Yeah. So how does MSC's therapy actually work? So for this MSC cell, this one cell, it will actually secrete a lot of cytokines, growth factors. There's also this term as extracellular vesicles. So these are actually secreted proteins and so forth, which will actually target different areas. Like for example, it can help to reduce inflammation. It can help to modulate immune cells, and then fight cell death, and then promotes wound healing, things like that. And of course, it can help to regenerate the tissue by stimulating the local stem cells to regenerate that disease area. And of course, this is the portion where I touch on the differentiation capability. It can differentiate into multiple tissues and be used therefore. And then of course, cell renewal is one of the important factors here. So one example of osteoarthritis, if you just use the traditional methods, it's I can give you pharmaceutical drugs to target your disease area, your cartilage defect, and I promote the growth of my cartilage again. But then for cell-based therapy, we use MSCs, which can differentiate into chondrocytes, and then at the same time secrete some factors to promote wound healing, reduce the inflammation, reduce this pain at the knee joint, and then of course you can repair your knee defects. So third portion, I'll go into the manufacturing of mesenchymal stem cells, how it's actually done in the industry. So for stem cells therapy, most of the time you need a large amount of cells. For autologous, just from the single patient, it can range from 10 to 200 million. And then when you go out to allogenic, you will need much more cells. It can go up to billions. So from just one umbilical cord, you get less than 1 million cells. You get less than 1 million cells. And then you aim to actually culture them in adherent conditions and then to grow them out into more for therapy. So to touch on industrial wise, this is actually what we do. We will get our cell source either by, for example, from here, this is fat source, liposuction. We work with clinical doctors to get these fat cells. And then thereafter, we will bring it to the lab. We'll isolate our cells, bank it down. Sorry, there's also the isolation and purification step. So we just get our stem cells and then we'll bank it down and store it. So every one of this vial is considered a master cell bank or a working cell bank. When we want to manufacture it, we'll just take one vial and then we will expand it in cultures and bioreactors. I'll touch on a bit on bioreactors later. Those are then harvested and then processed, purified and processed so that I can store it down into one vials for distribution. So at the same time, quality checks are very important. So in the industry, we talk about, is this still our stem cells? What actually are we going to introduce into your body? So this is one of the very important criteria that has to be talked about. So packaging and shipping is not so easy for stem cells. They are not like your typical drug where you just bring it around. They have to be stored in liquid nitrogen tank at negative 196 degrees Celsius, very low temperatures. If you see COVID, like Pfizer, their drug has to be shipped at very low temperatures, negative 80. You will also feel that stem cells has the same problem. Cold chain supply is a very important, obstacle for our industry therapy. And then finally, once it can reach the hospital, you can administer into patients and see and treat different diseases. So in manufacturing, we break it out into two portions, upstream, process, and downstream. So for the first part upstream, it focused on expanding these cells. Most of the time, you actually see 2D cell culture in T flask. This is what you actually grow in the laboratory. It's very small scale. You can get maybe 10 million cells at one go. But of course, in the industry, we need more than that. So we actually bring these cells from a T flask. We put it into small bioreactors. For example, what we are using here in Esco, we use cell cradle. And then this is good enough for very small scale production. Over here is just the harvester that we also use together with the bioreactor. And then thereafter, there's larger bioreactors out there. And at the same time, we use these isolators. We call it cell processing isolators, where we keep everything in a sterile environment, prevent contamination. And then we do this scaling up for industrial manufacturing. So bioreactors is the important term in industry manufacturing. So downstream wise, firstly, to expand, there's the upstream. Downstream is how you actually process to purify, and then to be able to fill it up into your final product. So harvesting, after we harvest the cells, we'll have to clarify for any solid waste. So for our cells, we actually culture them on small carriers, and then we grow it in the bioreactors. We have to get rid of this carriers. We need to go through this clarification step. Thereafter, we do some concentration, or we just wash the cells with buffer. And this will next go into filling, where we fill into vials or even into needles and syringe. And then control rate freezing before storing it in very specialized tank, the liquid nitrogen tanks. So some important factors of cell culture: pH, temperature, dissolved oxygen, agitation. I'll touch on it later. So pH, our physiological pH is actually 7.2 to 7.4. Temperature, I believe everybody's body's temperature is 37 degrees. You need to breathe, you need oxygen. It's the same for cells. And of course, you need to eat, you need nutrients. You need to have good quality nutrients. You need a balanced diet. You don't want to be having snacks all days. And then how do you actually get rid of this waste when you produce it? So this is also the same thing in cell culture expansion. So some of these are controlled by bioreactors, which I'll touch on it a bit. So there's many types of bioreactors out there in the industry, all meant for large-scale culturing. So very first, the T flask, which is the one I actually said is more used for laboratory scale. But of course, you can have tons of this T flask. People have used a multi-layer flask, but it also means that your process is very manual and laborious. So although there's this such of mono-layer culture system, most companies actually don't use this. And secondly, I have this list of different companies or the bioreactors out there. If you are interested, you can just take a closer look. But then just the principle, for example, this third tank bioreactors, we have our carriers. We just grow the cells on these carriers, and then we just keep doing stirring. This stirring will actually helps to allow the media to be mixed well. There's this constant pH, temperature, and oxygen supply from outside. And then you mix your media well, the cells get your nutrients and all they need. But at the same time, the agitation is a very tricky thing. If you agitate too much, you actually cause shear stress. And then, so this is where you actually need to balance out for the stir tank bioreactors. Therefore, people have went on to use rocking bioreactors. You just rock it gently like a baby. The agitation is lesser. There's lesser shear stress to the cells, but at the same time, if you realize it needs a larger surface area when you rock. The next one, hollow fiber bioreactors. This is when we have this kind of a new technology, hollow fibers. Cells are grown on the hollow fibers, and yeah, no problem. Oh. Okay, yeah. Let's proceed. So hollow fiber bioreactors, there's this hollow fiber technology out there. Cells actually attached to it. But if you realize that one bad thing is that you waste a lot of media. Media comes in from here and then media goes up. you actually use up quite a lot of media in this process. And then I have to say stem cells media are very expensive because they are just very fragile and they are much more critical compared to other cell types. Yeah. So this wastes a lot of media. And then at the same time with this, you can see there's a gradient flow. Over here, you get a lot of nutrients for the cells, but at the end, you get lesser nutrients. So it's not very consistent. And then last one, there's this fixed bake bioreactors. Your cells are fixed right in the middle with the carrots. And then there's a agitation somewhere to have the constant mixture of media to get your oxygen, your pH temperature all consistent. But of course, there's different pros and cons in each bioreactors. So at the industrial level, we need to ask ourselves, which one is the one most important criteria, or what does the cells actually requires? And this is where our bioprocessing knowledge comes in, to see which kind of bioreactors is suitable for cells. Yeah. So it's lagging. Okay. So again, to touch on why is it difficult to scale up cell culture is mainly the limitation to oxygens and nutrients. And then, for example, if we have a matrix bed, oxygenation can only be made outside it. And then there's sometimes this nutrients and oxygen depletion along the different bioreactors bed, yeah, compared to conventional types. And then there's also a presence of the shear stress that I touched on for the agitation for the stir tank bioreactors. So this can kill cells and increase cell debris in the medium, causing lower cell and product yield, and then increasing downstream causes. Yeah. So quality control management, one of the things that is important for industry. We need to make sure that we have enough of the cells, they are viable, they are alive. I'm not just injecting dead cells into your body and hoping that it will work. Identity, what are we actually growing? We will do specific markers, things like that. We will check that this differentiating. So this is where we do identity and purity checks to see they are what we want. Potency, we do assays to verify that it does work in the body. Like for example, I said it should treat osteoarthritis, so it should aim to reduce inflammatory cytokines. So this is where I can do immunomodulation assays. I put some stem cells together with my white blood cells, T cells, and then I say, okay, the T cells activities are lesser. Impurities, I need to get rid of all my carrots. I need to remove all my media. I don't want to inject them into a body. Microbiological safety, sterility. Is there any bacteria in the vial? I don't want to be introducing this to the body too. And then tumorogenicity. This is quite important, especially in stem cells, because like I say, stem cells, they proliferate. So there is chances of tumorogenicity. I can inject this and the next day, maybe you'll be getting tumor, you'll be getting cancer. There's this controversy with iPSCs for now. That is why it's still currently not in cell therapy yet. So the future of stem cells. You must remember that stem cells has any possibility to become all types of different cells. This is true, but stem cells are not a magic cure for all disease. The long-term therapeutic properties of these stem cells are still being investigated. And then for now, I would say that viable treatment is mainly on the immune portions, chronic inflammations, and then some of the other diseases out there. So it is not a magical cure. But then because there are aunties out there who goes to Malaysia, okay, can I have some stem cells therapy? So that is why FDA are also trying to fight this kind of controversy with stem cells. In the end, a lot of times, mesenchymal stem cells are not called stem cells anymore. They are rebranded as mesenchymal stroma cells. We don't want to give the wrong feeling of that stem cells, it does everything. So if you see mesenchymal stroma cells out there, it's actually the same thing but rebranded. So mesenchymal stroma cells, this is what I did mention, what they can secrete, some of the things. So there are new kind of therapies that are coming out, not using cell-based therapy, but trying to take all these small molecules or secretome to do the next therapy. This has lesser risk, I would say. Stem cell, no matter what, is a live cells. There's still this risk when you introduce it into your body. So people are moving onwards to, like for example, extracellular vesicles, proteins and peptides. You can do some engineering on these small molecules. Is a next area which we are looking into also. And of course, there are other things like genetically engineered stem cells, differentiated stem cells. And then, of course, a lot of people are using it for cosmeceutical benefits. If you go into Guardian, Watson, maybe you will see there's this stem cells renewal skincare. Yeah. Okay, yeah. So genetically engineered stem cells is one of the hottest topic for the future of stem cells right now. So it's actually when you have cells and then you derive from the patients. Derive from the patients and then you insert a gene carrying this you insert a vector carrying this gene that you are interested in, and then you put it into these cells for gene therapy before infusing back into patients. So there are quite a few examples out there for now. This is for example, T cell CAR T treatment. So you can actually take out your white blood cells. It's not just limited to stem cells. You can also use it on white blood cells. So T cells, and then you do a bit of genetic modification, and then you can insert back into the patient. So in 2013, there has been examples of leukemia being treated. A little young girl was treated with this treatment. So that is when I would say cell-based therapy are quite hot right now. Yeah. So the expectation of this stem cells market is expected to rise. By 2027, there should be a 15.1 billion global market saturation. So all this is just the projection of the market size. And then, of course, it's not just limited to stem cells. I would say cell-based therapy, cell and gene therapy is actually one of the newest area that is being focused on right now in the frontiers of the technologies. Yeah. So what I touched on just now, iPSCs, induced pluripotent stem cells. Again, taking a cell from your patient, I take a skin cell from my donor, and then I can reprogram it. I can take it actually from skin cells, from our blood cells, things like that. I reprogram it, and I have my cells. This can then be edited to do a correction of this mutation. And then I can differentiate into my heart cells, my microglias, my astrocytes, brain cells, and so forth. And so all this is part of what is going to happen in future, genome editing, and then differentiation into different cells before putting back into patients, or we can also use it for drug screening or other understanding to diseases. So this is the next generation where immune-induced pluripotent stem cells will be done. Yeah. So how far are we actually right now? For now, there's been lab-grown heart muscle cells from iPSCs that's been transplanted into a human. So this happened in 2020. And then, of course, there is a woman to receive cornea made from this reprogrammed stem cells. So this also happened in 2019. So these are one of the first transplantation using iPSCs. And then, of course, over here, you can see there's this mini human heart model that's functioning. It's functioning in the lab, but for now, there's no real studies or transplantation using this kind of a human heart yet. But this is what we hope to see in future when we can really use induced pluripotent stem cells and cure disease or do organ transplantation. Yeah. So that's the end of my presentation. I hope everybody had fun listening to this stem cell therapy. And then, yeah, if you have questions or maybe later. Yeah.
Okay, thanks Geraldine for the talk, very interesting. And now we're going to talk about the next talk will focus more on bioreactor and fermentation. So we're flipping the order again, but Geraldine just spoke about the stem cells. Now we are looking at microbial fermentation and bioprocessing, and then followed by the sharing by the CEO on how he actually started all this work in his company. Okay, I'm gonna have Mridul come on here now. So there is a question here, potential of recipient's immune system rejecting the stem cell.
Yep, so I can answer this very quickly. For mesenchymal stem cells, they actually have a low tendency of this immune system rejecting the stem cells, because when we talk about rejection, it's certain markers on the on the cells. So for this mesenchymal stem cells, there is no such existence of this marker. So it's actually HLA, DR, and some surface proteins to cause this immunogenicity. So I would say for mesenchymal stem cells is relatively safe. And then induced pluripotent stem cells, so autologous, if you are injecting back into yourself, into the same person, then there's no risk. For iPSCs, if you are injecting to the next person, so this is where genetic modification can come in. So it's not so ready yet when all our scientists are actually trying to design these genes to remove all these markers on the cells, such that we won't cause this immune rejection. But of course, in future, there'll be the chance that there won't be this kind of immune rejection. Yeah.
Hi, I've got a question about stem cells.
Yes, please
Hi,
go
so
ahead.
my name is Irvin, and I'm a year 2 bioengineering student. And I was just wondering, because as you were talking about the stem cells, you mentioned that stem cells had the potential to differentiate into many different cells and to secrete many different molecules, right? So I was just wondering, as we introduce stem cells into like different individuals, how do we control the differentiation into like the specific cells that we want to end up with?
Yes, so this is a very specialized area for the differentiation. For mesenchyma, it can differentiate into 3 types of stem cells. It can differentiate into 3 types of cells: osteogenic, chondrogenic, and adipogenic. So this is actually well studied by using media. So when we culture cells, we use media. So if you add different growth factors, it can actually stimulate the cells to grow to a certain lineage. So this is studied using small molecules, and then we try to add different kinds of these molecules into the media and see how they actually differentiate into the various lineage. Yeah.
Thank you.
So for now, I'll say that mesenchyma stem cells, it can only differentiate into 3 lineage, but like pluripotent stem cells, the heart, the cornea, the neuron, the skin cells have been differentiated into. Liver cells, kidney cells, people have done some studies, but then not all are very well understood. So even if I say heart, it comprises of many different kinds of heart cells, the cardiomyocytes, the atrium cells, the ventricular cells. So differentiating into heart cells, I can differentiate into some type of heart cells, but maybe I can't control the percentage of my cardiomyocytes. So this is also another tricky portion happening in the stem cells research world. Number 1, to differentiate into what we want. And then number 2, to be able to control this percentage of cells that we differentiate into. So just something to share.
Okay, I hope that explains it. And now we have Mridul on the top.
Hello everyone. Thanks, Prof. Lim, for inviting us to share our research and our technologies here with everyone. So yeah, screen. Okay. Yeah. So today I'll talk about industrial fermentation technology and the microbial bioprocessing that we use in the industry mainly. Okay. So I'll talk about more from more theoretical point of view what is actually fermentation means. So fermentation, the word actually came from Latin word, and it means like to boil something. So boil is like it's mainly used for like to act the yeast on any fruit extract initially to produce carbon dioxide from in an anaerobic process. So later in different industry it has different meaning. Like in biochemistry it means like to generate energy from catabolism from of organic compound. In biotech industry mostly we use this term is like to produce different kind of biologics by using cell culture in doing the mass culture of microorganism. So I'll talk more about the biotech industry, in the biopharma industry, what kind of product we produce here. So in the fermentation, there's a diverse application of this one, like pharmaceuticals, then food ingredients or pharma ingredients, and also in the cellular agriculture area. Like in pharmaceuticals, like we produce different kind of recombinant product like proteins, enzymes, peptide, antibodies, like that. And then food ingredients like pharma ingredients like plasmid DNA, also we produce from fermentation. Then different media component like growth factors, taste enhancer. These are more like food ingredient we can produce from fermentation. And the new area is coming up in this like cellular agriculture, where lots of alternative protein are produced by fermentation for mainly for in dairy product, then meat product, and also single cell proteins are produced by fermentation. So this is a very new area in biotech industry. So if I go show you the chronological logical development of fermentation process from like very early age. So like P90s, mostly fermentation was used for making hard liquor or like vinegar to preserve food and everything. Yeah. So that time it was that there's no pure strains or used, but the mixed strain or used for fermentation. Then moving on from there in 90s and early 90s, like then batch and fed batch processes are introduced, and also like some pure microbes are used for producing different alcohol, and baker yeast was used for making food ingredient. Then moving on from there, then when penicillin invented, and penicillin amino acid enzymes are also used, then the technology of mutation also used for selection of particular strains, and more like pure strains are used in the industry. Then coming to 19 in the mid 20th century, then single cell protein are used for hydrocarbon and other live cell stocks. Like genetic engineering was introduced at that time point. Then pilot scale production also started. So you can see the change of the fermentation. The vessel is changed from like from wooden drum towards like stainless steel, and then become more efficient in like 1980s, like when the sensors are introduced, and more control system has been introduced to make the process more controlled and efficient. Yeah. Then early 2000s, and then presence is like genetic engineering, and then synthetic biology is like the technologies are more improved and more. So those are also applied in the fermentation technology. So now we can engineer we can engineer the particular strain. We can engineer them with a particular product, and then produce them in large scale. And we use this technology to establish the previous fermentation process also. So, to do do fermentation, the microbes are really important. So, if you can see the more approved product in the market in 1980s to 2010, you can see the increase the number of product that is approved in the market is produced mostly in microbial cells using E. coli or Saccharomyces cerevisiae, so which is the large in the number, and the second is like mammalian cell, and then others like insect cell and transgenic cells are also there but very, very low in the number. But mostly mammalian cells and the microbial cells are the main expression platform or for fermentation. So I'll go to several microbial cells that are mostly important in microbial industry, fermentation industry. So E. coli, which is actually considered as the workhorse of biotech industry. So E. coli we use mostly because of its rapid growth in very inexpensive media, and it can be growth with like very high cell density. The problem with this E. coli is like that it can produce a lot of high endotoxin and host cell protein, which need to be purified, and also it has lack of post-translational modification function, so which actually make it more difficult to use in for human proteins and others, and also for large protein like more than 60 kilodalton, it get insoluble in the body, and then it's difficult to do the downstream processing and purification. So still that in the industry, more than 30% of the approved therapeutics agents are actually produced in using E. coli. Like big companies like Boehringer Ingelheim, they use microbial fermentation since like 1980s, and then other companies like Sandoz and LifeTech, they develop different strains that to be used in industry for expression of different product. So this another one, Pichia pastoris, which is a yeast cell, so which is the methylotropic yeast. It also can be grow in very high cell density with very low or inexpensive media, and regulatory authorities are very well known about this process. So it's like when you use this expression platform, it is very easier to get through the regulatory process. However, it also comes with some disadvantage, like it use methanol as a inducer to express most of the protein, and then it also require a lot of oxygen supply, and it causes the increase of the temperature in the fermentation process, which also needs like extra energy to reduce the temperature of the process. So altogether is the pros and cons are there, where this one is used in production of vaccine and antibody fragments, and in different industries. Saccharomyces cerevisiae is another one, which is yeast which is used very commonly is known as Baker yeast and used very commonly in industry. The difference between this Pichia pastoris and Saccharomyces cerevisiae is like that Pichia can do the glycosylation, which is like a more optimized, then Saccharomyces cerevisiae does hyperglycosylation, which is very which makes this more difficult to work with in for human proteins and other things. And is also used for different biologics production like insulin, hepatitis B vaccine, and antibody fragments. Other potential microbes are there, like a Fusarium venetonum. This is like used for single cell protein production. This if you can see some product in the market, like I think I forgot the name of the Chiew, I guess. This one produce this product for selling single cell protein as a food ingredients. Yeah. And then Pseudomonas fluorescens, this one used by Dow company for expressing different proteins, is a parallel expression platform of E. coli and Pichia pastoris. And also Lactococcus latis, this one is now very famous in larger scale production for heterologous expression proteins, and also considered as like viable choice for membrane protein expression. So yeah, so these are the few microbes that are interesting in biotech industry. So moving on to the how we actually process these microbes in our bioprocessing in industry. So is an overview of workflow of microbial bioprocessing. Like I divide this into like 5 different sections actually. Like when the first section where we develop this strain as for expression and for production. And then we go to upstream development, where we do the fermentation, then downstream purification step, and then at last quality control, and then storage, formulation, storage, and shipping. So I will go through the details of this one in the following slides. So I'll give you a very overview here. Like in the strain development stage, we select the product, which part we're going to express, and then we select the organism, which organism is actually should be used, like E. coli or yeast, or like bacteria or yeast. Then we can do the strain optimization by doing some mutation on that, and to find out the best strain that that will grow the cell, grow in numbers in terms of biomass, and also express the higher yield of protein. And then we also get the in parallel we get the gene of interest, and do the cloning and transformation to get the final version or final strain of this microbes. And then we can do high throughput screening to optimize the fermentation condition, and then we prepare the master cell bank, and then do a small scale culture to get the working cell bank, and then final, later we do benchtop or like a large scale production in fermenter. Yeah. So from there, downstream processing is like more like cell lysing, then filtration, and then purification, polishing stage in chromatography, and then there's a parallel process for the quality control. It's like in process or like after production. So it's like this one is like very simple picture here, because I cannot show a lot of quality control issue in this small figure. So yeah, so moving on, there is like the formulation step, which is like a freeze drying, whether the product is like a dry product or is a wet product. Based on that, we decide whether we do it like freeze drying or directly vial filling in a isolator in a very aseptic condition. And then moving on from there, labeling and packaging and shipping. So that is the overall microbial bioprocessing. So I'll go through several parts, like in the following slides, like so how we do industrial strain development in our Esco Aster. We use this plasma technology, atmospheric room temperature plasma technology, to do random mutation on the cells. So in this process we can produce a lot of mutated strain in a very shorter operation time, and because it's a random process, it produces a lot of different variation of the strain of the mother strain. From there, we do the screening by another technology is called microbial microdroplet culture system, where we can really sort out these different strains based on their growth, and also based on their expression level. So if you can see this figure, like here is like just a simple growth curve with this is a mother strain, how it grow with the time. We can measure this, like we can run like at least 200 parallel fermentation process, and we can measure their growth in real time. And this one is showing the stability of the strain with different passage and different generations. And then this one is like to optimize the one factor optimization is called. This means like when you want to know, okay, any particular media component or particular say in induction in the, like how you induce how much glucose is needed for optimal growth, you can optimize that particular one factor using this method. Like you make a gradient of this particular media component, and can see the growth in different condition in this machine. This machine is actually based on the microfluidics, so it can produce like 200 droplets. Each droplet is work as a fermentation system, and then it can show you the results at the end. Yeah. So if you want to know more about this process and everything, we have a poster in our website. If you can go through this link, you can find that more information about this one. So molecular biology, I think, is like a very old technique, and everyone knows this thing. So it's like we get the gene, and then we do molecular cloning, transformation, selection, and then we produce this cell. We do the cell banking for getting a strain for expression and production. So I'll go to fermentation. So fermentation is like actually start from the cell banking, the stock culture from the cell banking, from their seed culture preparation by shaking flask, and then seed fermentation, and then production fermenter. So how this is how a fermenter, industrial fermenter looks like. It has like different components, like first of all the vessel. So vessel, and then the vessel comes with mostly temperature controlling water jacket or a bottom plate. So to control the temperature, it also has this agitation, the motor shaft and the impeller, with the impeller blades to maintain the agitation in the media, in the vessel. And then it has lots of sensors, like pH sensor, temperature sensor, and then like anti foam sensor, to maintain and the control the different parameters. So usually the control parameters are like the media and the feed flow rate, then temperature, pH, oxygen, agitation, the substrate concentration, and osmolarity. These are the few parameters that are important to control in the bioreactor. So to design the bioreactor, there's other parameters are very important, like the operating volume. First of all, how much volume we use for the designing this bioreactor, then the height and the diameter ratio of the vessel, then the impeller, what kind of impeller we are going to use for different kind of microbes based on their how much shear stress it can take. Then the mixing ratio, then oxygen transfer coefficient, which is very important for designing a bioreactor, that will ensure like how much oxygen we can provide to the cells, and then power ratio to maintain the how much power is actually required per volume of the per unit volume of the bioreactor. Then how we operate this bioreactor, there's a several method of operating, like different modes, like batch, fed batch, continuous, and perfusion. Perfusion is actually mostly used for mammalian cell culture, but if for microbial, we use this batch, fed batch, and continuous process mostly. In the batch process, basically you add all the media and then the required component in the vessel, and just do the fermentation, run the fermentation using your specific condition, and at the end you harvest everything. In the fed batch, we feed the media component and time to time in the vessel, and the volume is actually variable in this case. And continuous process, we feed and also we harvest at the same time at the same flow rate, so that the volume remain constant in the vessel. So again, these processes are like for industrial scale, sometimes the yield is not good enough in batch and fed batch process. So we do repeated batch process. In this process, we follow the similar way of, we start the process like a batch, but at the end we harvest like 80 or 95% media, and we refill with fresh media. So that will increase the and run it for several rounds, so that we can increase the yield of the production. So yeah, moving on to the downstream bioprocess. Downstream bioprocess for microbial is like very dependent on the what is the location of your product, like whether it's an inclusion body or it's like secreted outside of the cell. So based on that, the downstream process vary a lot. So we need to consider that, like when we are selecting the downstream processing, the concentration of the product, the degree of impurities, the acceptable standard of purity of the product, then also the application of the product, and then price and everything will come into play when you are selecting the different processes for downstream. So these are the very basic steps for downstream, like cell followed by fermentation, like cell harvesting, cell lysing, inclusion body solubilization, clarification, and purification. So in the following slide, I will show a few of the techniques that we are using, our machines we are using for downstream processing. So for harvesting the cell and or the clarification of the lysed cell, we use mostly the tangential flow filtration system. So this system is very efficient in separating the cells from the media, and it's a closed system. So we can use single-use materials for doing this operation. So there won't be any chance of contamination between the different product. So the reason this is like very interesting, because the the flow of the feed and the permeate is like they are not in the same direction. They are like 90-degree. It's like a 90-degree angle. So there's no chance of membrane fouling or like clogging of the membrane, which makes this more efficient in process. So the in this process also we can like different kind of membrane are used, like membranes like say you need a very high surface area, you can also get it by using this cassette kind of filtration. So it will compact the large surface area of the filtration membrane in a very short space. And there's a different kind of cassettes are available in the market. Like if you want to do like sell for harvesting, you can use this large like microporous is a large pore size. And then for clarification, you can use like different pore size, like which is like 0.22 micron and everything. And you can go like molecular cutoff filter up to like 3 kilodalton also using this. So other systems are like that, like centrifugation, which is very classic for using in lab scale, but industrial scale is very difficult because of it's expensive, and also continuous processes are very hard. However, in the market right now, there's several technologies are there who can do single-use centrifugation, and also like continuous centrifugation using this kind of centrifuge machine, where you will feed in the you put the feed in the middle, and then there will be a positive displacement pump to remove the solid part or the slurry part from the centrifuge, and you can get the clarified portion also in the outside. Yeah. Another way is of doing harvesting or clarification is like flocculation, which is mostly used in food or effluent treatment plant. So these are like more doing flocculation using some different kind of flocculation aid to reduce the to make the cells more neutralized, and then other than and then reduce the surface hydrophobicity, hydrophilicity to make the cells precipitate in the down, so that you can flocculate. So and then cell lysing is another part. If you need this when your expression is inclusion body, then there's several techniques like physico-mechanical techniques and chemical method, like to break the cells. But mostly used technique in the industry is like high pressure homogenization, where you are breaking the cells using very high pressure. So you are putting your cells in a very high pressure, and then release the pressure in a very suddenly, so that there's a cavitation happens. So because of the sudden release of the pressure, so this cavitation actually breaks the cell apart. So when you are selecting like you're doing the homogenization using different cells like bacteria or yeast, you have to select the different pressure based on the strains, and then you have to optimize this pressure. And because this pressure is important, because when you increase the pressure, is a high chance of breaking the cell, but at the same time you are increasing the temperature, so which is not good, may not be good for your product. So we usually use low pressure, and we run these things several rounds and several cycles to get the 100% efficiency in cell breaking. Another process is like a chemical process, alkalization, mostly used in producing plasmid DNA. So we use like a alkalization solution and then neutralization solution to break the cells, and then keep the and then extract the product like a plasmid. And this is very expensive process, but it's required for very specific products which require very low shear stress, and then yeah. So we use for these techniques. Yeah. So at the purification and polishing, mostly we do with the chromatography for pharmaceutical product. So we in Esco we use the biorad process for process development, which is go up to like 6 liter per hour flow rate, and then manufacturing scale we do like very low scale to like very high scale, like high flow rate like 600 liter per hour, which also comes with single use accessories, so that you can run different product one after one without any contamination happens in between. And okay, I'll finish quickly. How many minutes? 2? Okay. So Yeah. So a formulation is like it depends on the what kind of product we are making here. So this is several kind of formulation like drying techniques if we want to make the dry product. So this could be like surface contacting process, or could be a surface non-contacting non-surface contacting process like spray drying or freeze drying, or for surface contacting is like rotary drum dryer, which mostly used for food ingredient or food production in cellular agriculture process. And non-contacting like freeze dryer or spray dryer can be used like in pharmaceuticals. And at last, the peel and finish process, we maintain the grade A environment for bulk filling or vial filling of liquid product. And then we have like different kind of isolator like cell processing isolator or turbulent flow aseptic isolators. Those are like used for filling the vials or filling the bulk filling into bags. Yeah. So there's a glimpse of the Esco's bioprocessing site that we have in Singapore, and also in our partnering sites. Like here, we have small scale like for process development to up to 10-liter bioreactor, then 50 to 100-liter bioreactor, which has stainless steel, and then up to we can go up to 50,000 liter in our with our partnering company. This one, and yeah, so is it like a for upstream, and for downstream we have like these technologies like ultrafiltration, homogenization, centrifugation, dryer, evaporator, and chromatography for to go up to like industrial scale. And these are the mainly we use for process development in lab scale. I think that's it about the bioprocessing for me.
Okay. All right. Thank you, Mridul. That was my student who is one of the first actually developed these kind of processes in my lab, and then now he's doing it for the company. So we're really happy to have him here today to share what he has. Now I have, while we are setting up for the next speaker, the CEO of the company, I'm going to fill up the gap with a little bit of announcement. I'm chairing the let me see, let me grab this announcement for a Women in Synthetic Biology conference, and then we would like to invite you all to submit an abstract if you are working in any places close to engineering of biology, biological cells, and not biological cells, but engineering of microbes or whatnot for synthetic biology purposes. So the link to the symposium is on the chat box, so please feel free to check that out. And if you would like to see what it is and you have questions, feel free to check them. And here's Costas from Synergy. This particular conference or symposium is hosted by Synergy and ASBA. ASBA is Asian Synthetic Biology Association. And Costas, would you like to have any further announcement?
I would like to say hi. So hi everyone. Can I share a slide? No. Yeah. Okay. So can you see? You should be able to see my slide. Yeah. Thank you, Shireen, for the very nice event. I'm happy to see you moving on virtual, on hybrid events. Hopefully soon we'll move to only physical. Yeah, so we are very happy to have this Women in Synthetic Biology Symposium together with Synergy, Asian Synthetic Biology Association, and the local communities. So I would like to invite everyone to join. this symposium. Please, the more junior researchers, please feel free to submit your abstracts. You don't have to be a woman or Asian to participate, but it's a good opportunity to highlight the research done by our communities here. Synergy in general has more events. So if you want, please feel free to look what we are doing on our social media and join our other Synergy webinars and discussions at the moment. And hopefully, as I said, will be physical.
Okay, thanks so
soon.
much for us. So we are going to now continue with our speaker. And this is the CEO of the Esco Aster, and he will be presenting to us and then share how he actually started this company and what inspired him and how he actually make it work here. Okay.
I hope it's working. Ken, so good morning, everyone, both who are here in present and still connected virtually. I know this is a very soon time for lunch, so I'll try to be short, but may overextend a bit. Thanks, Paul Sim, for inviting me. It's my pleasure to be here along with colleagues. Right. So I'll just share with you more on the journey of the company as a whole, and then personally myself as well, right? So I've been in your shoes before. How do I move slide? Okay, so I've been in your shoes before, studying, and okay, some of you have already graduated, but you know there are always choices, right? Once you graduate, right? So there are different things you can do, from doing another undergraduate degree in case you realize that you're not so interested in science. Some people may jump straight to masters and do a PhD under a professor. Some then do a double PhD if they really like to get permanent MH, or if they really like the subject of different fields in a way, or there are people who have serial PhDs in a way. Some gain work experience and then join a spin-off. Some gain work experience and join the industry, like Mridul did. Some gain work experience and then do postgraduate studies. So this could be a master's or MBA, become a doctor. So it's more common now in Singapore to have course undergraduate science or engineering, and then they have some scholarship program to do postgraduate as a doctor, right? There are a few medical schools that do that, right? Or gain work experience and then move directly, skip masters and do a PhD, right? So there are actually many choices in a way. So I'll just share with you what I did then, right? So I studied in Victoria School. So then I went to VJC, like most Victorians did. I went to University of Sydney. So I did, but I explored studying many different things in a way before I chose life sciences, right? In a way, actually my main major is in clinical psychology. So, but the Australian system until now, where like in NUS psychology art school merged with science, in Sydney is a postgraduate as a doctor. So actually, basically I studied neuroscience, right, in undergraduate. So it was all science. There was very little counseling because it's Australia. So as part of my thesis, we made mice addicted to beer. we did funny things in a way with neural stuff in a way to understand how all these drugs work in the brain, right? But it's a very fascinating space, and it's still a very fascinating space even up to today, right? Because chemically, in a way, I don't know whether you all have loved ones or close ones who are clinically depressed, but people who are depressed, when they take the medication, chemically speaking, it works within 10 seconds, right? But they're still depressed, right? No matter what you do, you can't get them out of that slum, right? In a way, there is some like a physical soul thing going on, or maybe, right? So it's still a very fascinating space, because biologically things work, right? But the person is still depressed. So, it's a very interesting space and field, right? Then personally myself, in all this journey, I realized actually when I was studying in University of Sydney, which is up to date, I still think it's the best time of my life away from parents. There is no control, there's zero curfew. I can do whatever I want, right? Basically, in a way. But then I realized that I like to conceptualize and create things, right? So I think it's actually very important, even as you're still studying now, to have a deep understanding of yourself, right? Because that would really inform your future life decisions, whatever it is, right? So for me, I realized that actually I like to conceptualize and create. I think I could have made still a very good clinical psychologist because I can create either drugs or treatment programs. Or I could have done something else, right? Even up to now, in my day-to-day spare time, I'm still writing poetry, I'm still playing drums, I'm still involved in music, I'm doing many other things. But the root of it, everything is that I like to create, right? So I think everyone should find your own root and what brings you joy, because like it or not, adulting is hard. It's a lot harder than you would expect. Yeah. So, and the other thing I realized for myself is that I like to do things that nobody else can do, right? And solve a very big problem statement in a way, which I'll explain later. But mainly it's about healthcare access, right? How can we reduce the cost of goods for all these cutting edge medicine? Some of it are stem cells, like what Dr. Geraldine shared. Some of it are biologics, like what Dr. Mridul shared. Also, I can live with a certain amount of uncertainty. So again, it boils down to your personality, right? Some of you can live with more uncertainty. Some want things to be very, very sure. And so maybe joining or doing a startup may not be the best idea for you, right? Again, to each your own, right? Understand thyself. And I can combine emotions, and I'm still a bit more happy-go-lucky, right? By nature, in a way. So yeah, so that's me, right? In a way. So then what do we build in the company actually? We have various manufacturing platforms. We are the first in class for cultivated meat to do contract development manufacturing. So we are the only company actually globally now with a food regulatory license. And we are making the first normal food approved product as well in Singapore actually. In our cell processing facility. We are the best in class for adherent cell culture, which Geraldine shared on our Tide Motion platform, which is also used for cultivated meat actually. And we are best in class for precision and continuous fermentation. So actually, before I started all of this, in a way, what was very important is and this again is quite similar to what Elon Musk said as well, right? But go back to the basic principles and have a deep understanding of all of the basic principles, right? So we are cell culture specialists, of course. So then we need to have basically a PhD in every single type of cell culture, right? But so there are different culture principles, and the main thing was to understand what was the root in all of these culture principles. What were the good points? How can we adapt and create systems to solve one of the biggest pain points, right? Which one of the biggest pain points actually is oxygenation, right? You can live without food for a few days, but if all of you just hold your breath, you will die from lack of oxygen in your brain, right? So cells are very similar in a way. How do we get more oxygen, therefore, to cells, right? So we have our Tide Motion system, which then through the ebb and flow of media and natural oxygenation has the highest KLA and oxygen transfer rate, right? But it was understanding all of these other principles that really help us to develop our system. And we are the only bioreactor adherent pack-based system with full online sensors and monitoring and glucose control that has automated cell harvesting in terms of a linear scalable platform, right? Most of their systems may have automated harvesting and pH, but they are you have to scale out. That means you have multiple units of the same bioreactor rather than scaling up. Or you may have automated harvesting, like in the case for multi-layered culture, or we call multi-stack culture. But these do not have a pH and DO monitoring within the culture vessel itself, right? So it's actually understanding broadly all of the fundamental principles in whichever field you're in actually would really help you to have a broader understanding on on developing things, right, in a way. So, this is just a slide to show the differences of from multi-layered culture to microcarriers, which Geraldine mentioned, to microporous carriers, which I won't go into too much details, but you can take a look. So at the root of it, therefore, we have our tide motion system, which is automated harvesting, and we can then scale out or scale up from very small to very large scale, which we're also doing with cellular agriculture. So we can change our carriers to an edible scaffold, and then we create meat. And because okay, I know the image is quite small here, but it's our matrix or pack bait system is cylindrical. So actually you put edible scaffold inside, it becomes kebab, right, basically. So you can take the whole vessel out and then shear it in a way, and then, okay, it's kebab, you can grill it away. So that's something which with our team we are trying to do. Okay, there's low battery. I hope there's enough battery. And so in a way, how did it work in terms of natural oxygenation? This is very much like swimming in a way, right? And we have the highest OTR. Of course, now it's Olympic season, and it's unfortunate that Joseph Schooling didn't perform the best he could, but pretty much we are top performer or Olympic gold medal for cell culture scale up for adherent cells, right? So we do this by having this tide motion system, which is pretty much like breathing, right? Or like swimming in a way. So when our tide motion goes up, that goes to media exchange phase, which the glucose or the nutrients feed in the cells growing inside cross-linked fibers. You can see here, image here. And when the media goes down, then there's natural oxygenation phase, right? So it's really like an artificial lung, right? It's really like swimming in a way. So because of this, we have we don't use any impellers for oxygenation. It's much easier to scale up or scale out, right? And in a way, we can use the same principle. And of course, we are still fine-tuning the system as it goes along, but we can use the same principle right from preclinical in our R&D in an academic lab, all the way to GLP, which is testing in animals, to then clinical trials, right? I think due to COVID-19 now, everybody is expert in either diagnostic and or vaccines, right? Everybody is like, oh my goodness, is this vaccine safe? What is a clinical trial? How is it developed in a way? So, but basically, it's clinical trials are development stages in which we test for safety, more or less in a phase 1 trial. In phase 2, we test for safety and efficacy in a way. In a phase 3 trial, in a way, we then test for safety, efficacy, potency, and a whole bunch of other things, and then we commercialize it, right? So we can use the same method from end to end in a way. And also it's because of this, like Dr. Geraldine mentioned as well, those Thai that go to Malaysia for beautifying or anti-aging is, I don't know what they're doing lah, in a way. It's maybe they have too much money, maybe they have, I don't know, but there's actually animal evidence to show when you inject stem cells intravenously, you cause embolism actually. So actually they're killing themselves, right, in a way. But okay, I don't know, maybe there's a certain price to pay for beauty, who knows. But in any case, that's why all those things don't make sense lah, right, in a way. So from our same principle in a way, we can scale up from a very small scale all the way to autologous or as a seed train or commercial platform. Again, too many words. In the interest of time, you can look through the system, right? But in any case, we have a direct scale up method using the same tide motion principle, and we are right now working on parallel bioreactors to parallelize it, so we can plan design of experiments, which is basically changing the conditions, right, in a way of what type of food, when to feed, what type of media to use in order for the cells or the viruses to grow. Then is how we do using our adherent system for autologous cell therapy, in that sense, in which we have steps from harvesting the cells to then thawing, we expand it in our bioreactor, we harvest it, then we do washing, filtration, and concentration using a tangent flow filtration. We fill it into a drug product that goes to control rate freezing, and then we freeze it in a way. So that's more or less the process, right? This is a good infographic to also explain, therefore, what we are trying to achieve, right? So actually, as a company, we have been around since 1978. Slides later show our history, but in a way, we have been supplying tools to the academic industry. And then we saw this very, very big gap, right? Which we call kind of like process value of death in a way. So there is a very big chasm of value to cross when you're trying to take something from the lab in a way, all the way into commercialization in a way, right? And so we realized therefore that there's this big gap in Singapore's ecosystem on how can we help all our PIs in that sense, like Prof. Savian and others as well, to translate actually all of their IP into something that's commercially used, right, in a way. So typically in an academic setting, in the labs, what they do is early stage discovery work, basic research, translational research, and then do preclinical development and animal studies, right? So in preclinical animal development, you're developing the animal model to test the efficacy of the drug, right? Or the drug delivery system, or it could be a cosmeceutical, or it could be nutraceutical, or any kind of complementary alternative medicine. Then when you go up to when you commercialize it, what we do is what we call good laboratory practice, or GLP testing. So this testing in a large animal model, which can be a rabbit, a non-human primate, or a pig, or a dog, or some other large animal, right? Because that is more similar to a human, basically, in a way, right? So this is how you know it's safe before you go to a phase 1 trial, in which it's a small batch of maybe 10 to 20 healthy volunteers, and you check whether the drug is safe, right, in that sense. Then, of course, you go to different peaks and valleys in the way of phase 2, phase 3 in a way, which many is also one of the hardest in a way, because many biotech companies fail as well, and then it goes into commercial, right? So there is a statistics of only 1 out of 10 biotech drugs ever gets commercialized, right, actually. So there's actually quite a high failure rate, actually. And we are here to fix this small little gap in this process value of death 1 in that sense. So as a company, mainly using fermentation and mammalian cell culture, we are broadly doing cells, viruses, extracellular vesicles, or antibodies or proteins or recombinant proteins, right? So these are range from mesenchymal stromal cells to vaccinia, to oncolytic viruses, which is another very big thing as well. Japan approved their first glioblastoma oncolytic virus. So this is using a virus to then kill the tumor cell, right? Which then is more highly specific, unlike using other chemotherapy drugs in a way. Because yeah, a lot of these chemotherapy drugs are actually non-specific, right? Even monoclonal antibodies, because some of the targets actually like HER2 and others are actually on healthy cells as well, right? So it will kill cancer cells, but it also will kill healthy cells at the same time. So some of these so oncolytic viruses, on the other hand, are engineered precisely to only target tumor cells. Then we have what we are going into, which is a cell 2.0 or 3.0 in a way, which is using exosomes or secretomes. So these are secreted out from cells itself. Before that, people thought this was junk or garbage that came out from cells, or like cell poop in a way. But actually, they are very, very valuable as a drug delivery system, and you can load drugs inside there. On a side note, actually, a lot of serum that comes from stem cells is basically cell poop, right? So if you're applying it to your face, then you're applying cell poop. So, but they have many good good effects in terms of yeah, upregulating or downregulating certain components in a way. Then, of course, we are using it for high-value diagnostics, antigens or subunit vaccines, antibody-drug conjugates, bites, trispecific antibodies, and we have drug bodies as well for single-domain antibodies. So, so as a whole, when I was looking at this, and I spent a lot of time actually in the developing countries. I went to slums in actually both Bangladesh and even in Philippines. And when I was a very, very young kid in a way, joining the company, it was a big irony, right? So you'll be staying in a 3-star hotel in the Philippines, and like you open up the one side of the window and you look at one corner, you see slums there in a way, and then you open the other corner, you see all these high-rise buildings, and then you're like, okay, what the hell is going on, right? It's like there is a very, very big rich-poor divide, and a lot of people that really do not travel to these developing countries really don't really see this, right, in a way. And and even up to now, every 30 seconds to a minute, there are about 40 kids, and even more so now due to COVID-19, that actually are dying due to lack of basic vaccines, for instance, right? So the main thing that really motivated me from day one was, how can we produce all of these drug products, including vaccines, at the highest yield, at affordable cost, make it linearly scalable, so that all of the work and experiments that even you do at various stage in the R&D and academic lab can be immediately translated to commercialization, right? So this was one of the main aims, right? So this information is actually also on our website, but then basically we are looking to reduce the cost of goods for each of these new modalities, whether it's a CAR-T for blood cancer, or whether it's stem cells, whether it's vaccines or animal health as well, because up to now still we don't know where COVID-19 comes from, right? I mean, people thought it was bad, then it showed all the cover people of like people in China eating bad soup in a way, which I don't really think they do, but yeah, it looks kind of gross actually. Then people thought it's pangolin in a way. Then people thought it's like minks in a way, because there's a COVID-19 virus in a way in Denmark, and then they actually like take off the mink fur for all the fish tai-tais, right, in a way. So it was there's a big fur bearing industry, and actually there's a COVID-19 strain there that they had to kill all the minks, unfortunately, and ferrets for that matter. So you know, so we still don't know where, right? But then actually by taking better care of animals, it affects us as well, because then it's a one world and one health, right? So cost was always also one of the things, right? So from that in a way, both Dr. Mina and Dr. Geraldine went through all the different steps, but this is our facility in which we are doing all of this cell line creation up to phase 1 and 2 clinical trial material. So then maybe a brief history then of the company as a whole. So Esco started in 1978 by my parents. They founded the company. They're both pharmacists by training. Graduated from then was University of Singapore, and then now it's NUS in a way. And we expanded the business as Singapore expanded as well, right? So in 1985, which is actually when I was born as well, late Sydney Brenner advised late Lee Kuan Yew to invest in life sciences. If not, Singapore remain a city of technicians, right? So that led to the formation of IMCB, which is one of the first research institutes in Singapore in that sense. And then as Singapore moved more towards life sciences as well, this is when the company as a whole moved more towards life sciences. In 2002, there was a patent of our tight motion bioreactor, which is a second single-use bioreactor after the Wave platform in 1998. And coincidentally, there was SARS pandemic as well. So we have been working on this for a long time, right? But then just to show a bit of the timeline and our journey in that sense, right? So from 2002 to 2010, when I joined the company, then we started working on vaccine self-sufficiency. In 2015, so 13 years after the patent, then we started to really internationalize and upgrade our tight motion bioreactors to really make it CGMP, right? Which stands for Common Good Manufacturing Practice. This will allow it to be sold and used for our USA and American clients and collaborators, right? And then we were doing PD at client sites, and I had a team of scientists with me in a way. In 2018, which actually our first Singapore NTU graduate postdoc joined us, which actually is Dr. Geraldine. So she's been with us in this very long journey in a way, when we were in a very, very small lab, even smaller than this room actually. I think it was 1/3 this room in a way. In a very, very small lab on the 7th floor in block 71 to level 3 in block 71. And then we are now in block 67. I think probably Dr. Geraldine has moved more times than she has moved house, maybe. So we have to panjia, which is Chinese for moving house, right? So we have to like panjia many times, right, in a way. So like Geraldine has really been a very tough soldier and been with us for a long time, and hopefully many more years to come, right? To really see us grow from from almost nothing to where we are, right? Which is kind of like Singapore as well. Then in November 2020, we are planning the larger scale drug substance and drug product site. Then now in 2021, we have our CGMP site, which is operational for up to phase 2 clinical trial material. You know, but actually such is life, and in a startup, many things change. And we have to change the circumstances as well, right? So we had to abort making the site in a way, and this in a way, caused some unhappiness in actually some of our new colleagues who then left the company, unfortunately. But we had to do so because the cost was just too high, right? So it's 40% higher due to all of this COVID-19 shutdown in a way to construct a site. So something that cost 100 million, cost 140 million for no other reason than shortage of manpower, shortage of materials, everything was short, right, in a way. So it just didn't make business sense, right? So actually, many bad things happen in a startup in a way, but you just have to adapt and change the circumstances, right? And it's not something that actually you can really control, right, in a way. And it's quite unpredictable, right? Which goes back to my first point of like, I can deal with uncertainty in a way. So then this journey to me makes sense for me, but it may not make sense to you, right? Or it may make sense for you to join a startup if you can't deal with it as well, right? So it goes different ways, right? And then right now we are still ongoing to plan to be CGMP audited by HSA in December 2022, and we're still ongoing to plan our bigger site, right? So actually this whole journey from 2002, when it was the first patent, to 2022 is 20 years, right? And this is a very, very long and painful journey in a way. But it's not too different also to what is out there for cell therapy, for instance, right? Which is a fourth pillar of medicine, or mRNA in that sense, right? So, you know, CAR-T, which really the first CAR design was in 1989 to when it was approved, which was Kymriah by Novartis 2017, is also a 28-year journey, right? Biotech is very long. So if you're in it for a long haul. If you're not in a way, then yeah, there are other areas which you could always contribute in science, but you know, it's a very, very long journey, right? And it's also a very long journey for mRNA vaccines, right? Now it's a husband and wife team in a way. When they started BioNTech, and when they started it as well, everyone was laughing at them, right? I mean, it's well documented and many stories of them, but everybody was laughing at them, right? But then they formed BioNTech, and in 2010 and 2020, 10 years later, they formed, I mean, this when mRNA vaccines were then commercialized, right? So, you know, it's all a long journey, actually. But the journey in commercialization also, this is a typical cycle for the inflection point, right? So I would actually dare say in 2002, we were too early in a way, right? There was at least in cell therapy, right? In the cell culture space for cell and gene therapy, we were very, very too early then. And we also picked on the wrong horse. So in a way, the first bet that we picked on was CHO cells for monoclonal antibodies. But then most of the CHO cells, which are adherent in the beginning, got bioengineered suspension. So then they didn't need, many of them did not need adherent scale-up systems, right? Then in 2010s, in a way, we were working on vaccines for vivo cells in a way. And that was very good. I mean, I'm quite happy to say that the time motion system is now used for we have the 8th commercial vaccine now, which actually is for COVID-19. So it's also being used for COVID-19 vaccines. But then still, we didn't hit the inflection point in which, you know, it became very, very big, right? And then I dare say now we are really at the potentially next big thing, which is cell and gene therapy, right? So it's using mesenchymal stromal cells, which now we know if you just take mesenchymal stromal cells as it is in a pure form without any type of engineering, it does work. There's some clinical efficacy, but not for everything. So what we're doing now is actually either using viral vectors or electroporation to transfer genes to modify primary cells actually to either secrete better exosomes or to have better cell therapy. And this is actually what our tight motion system can very well do, right, in that sense. So very much we are really at the inflection point in which we can really scale up very large, right, as a company as a whole. But it took a long time, right, 2002 until now. It's still 21 years, right, in a way. So, you know, there are other considerations as well, right, on what type of company do you want to join, right? So, and this again goes back to yourself and understanding yourself, right? Are you a builder in a way in the desert? Because pretty much the startup is, you have to do something from scratch, right, in a way. It's like how Dubai came up from nothing. It's how Singapore came up from having pig slums in a way, and we were all extremely poor to where we are today, right, in a way. There is a certain amount of understanding to see to watch things grow, right, in a way. And if you are geared up for that you really like to do it, then that's great, right, in a way. Or versus working in a very structured system or a company in a way, right? But unfortunately, the more structured a company is, which you can see on the comic on the right, the more systems they are, the more pigeonholed they become, right? So, which is quite common at MNC. Yes, everything is very structured. Yes, everyone knows what their role is, but you are given a job description and that's all you do day in, day out. And most of the time, you don't know why you're doing it, but you are told to do it in a way. Versus in a way, in a maybe more dynamic or unstructured startup, you are actually creating the system and you understand how it came about and what is the logic. Again, there's no right or wrong, right? But it's really up to you to make that decision in a way, right? Of course, having said that, in an MNC setting, most of the time they allow you to switch from different function. I think minimum it's like every 2 years, right? But of course, the other unfortunate part is like most MNCs in Singapore who come in from Europe or elsewhere, a lot of managers, okay, it's less so now due to COVID, right? Because it's difficult in getting passes, but a lot of managers are helicoptered in. So you reach kind of like certain glass ceiling in a way. And unless your manager above you leaves, you have almost no chance to get promoted, right? Unless you switch to another company. But again, to each your own, right? It's whether you want to choose to grow with a company or to work in a very structured company in which you're pigeonholed and things are very clear, but you're pigeonholed. So yeah, I would say there's no right or wrong, right? But actually, no matter where or what you do, or what you decide actually after you graduate, I think there is some sense in being contented with where you are or the decision you have made, right? There's always a saying that, you know, the grass is greener on the other side, right? But frankly, there is another saying that the grass is greener on the side where you choose to water it, right? So you can make a choice to improve your situation on where you are at and make a difference in a way, or you can complain, right? So again, it's your own personal choice in a way, but I think in whatever decision you have already decided to, it's really where you water it, right? In that sense. Having said that, of course, in a way, you know, so it's learning how to be contented in every circumstance you're in, right? Because things are difficult, right? In a way, and no matter what, since you've already made a decision, then the best you can do is to make it greener where you water it, right? To make a difference, right? And really to be a change maker. And that's only down to you as an individual to decide whether you want to do that, right? The company can have the best systems in place, but still it depends on you to operate according to the systems, right? So it's or down to you to improve the systems, right? As it's built up in a way. Having said that, you know, there is this kind of like a peak and trial in a way as well, just as it is in drug delivery. But you know, a lot of times in a way, there is this is quite common in everything, including even Singapore, right? So like when late Lee Kuan Yew envisioned the merger with Malaysia being like, you know, the great for the country and we would be much bigger and stronger. So there was this peak of inflated expectation, right? And then when we were kicked out the way in a way, then there was this huge drop, right? In a way. So it's this trial of disillusionment. And then it was very sad for a while. He ran quite in public as well, right? And then in a way, things get better in a way. And then he becomes enlightened and realized that, well, I can do it, right? In a way. I'm Lee Kuan Yew, right? I have iron in me, right? In a way, I will do it, right? I will make Singapore from nothing into where we are, right? So, you know, again, it's being contented and understanding that you may be disappointed even if you have made a decision, but it's being enlightened and what you decide to do with it, right? Yeah. So in the startup, actually, timelines are rubber bands, right? In a way. And I see this in everything in our situation, right? Things are very dynamic. We also have to move with the situation, right? But then in a way, because the journey actually in this past decade really has allowed me to grow personally and professionally as well, right? I would never imagine I could really grow so much so quickly, right? I was like thrown to Thailand in the first week of working and I didn't know anything about what I was doing, right? But I was just thrown there for 1 week to go and sink or swim, like, in a way. So it's what you choose to do with it, right? In a way, I could have complained a lot. I could have like, you know, I could have decided to quit in a way. I could have decided to go back to study in a way, maybe, right? But in a way, I allowed myself to be stretched, right? Both by the company and by myself as well. and my challenges around me. And this is even up to now, we are still facing constant challenges every day, right? And we are still being stretched in all ways, right? Whether it's by clients or collaborators or other people. And this is very true in a startup, right? You're really building the plane as it flies, right? So starting company is like, really jumping off the cliff, right? And then you assemble the plane as it goes down in a way. Of course, then the other thing is that when you're doing a startup is which part of the plane do you build first? And if you do it in the wrong sequence, then it may probably your startup will fail, right, in a way, or the startup you're joining will fail, right, in a way. So, but it's a very complicated thing to answer, right? Because actually in a startup, everything is happening simultaneously, right? You need to raise money for investors. You need to build operating systems on how and who to do what. You need to maintain the systems that have been built. You need to develop infrastructure in a way that and ensure that both explicit and implicit and the tacit knowledge is then transferred. You need to have human resources and training as well to attract, retain, and develop the existing employees. And then you need to develop technology to make sure it's ready for your next milestone in terms of getting unlocking either the next collaborator or reaching the milestone in terms of the technology to get it commercialized, right? So it's difficult in a way because actually when I was starting this, I had to do everything simultaneously, right? So it was difficult to manage and difficult to know what part to build, right? But I think the important thing is to make a good start, right? So don't make the first, don't jump off the springboard in a way, like the image below, on the wrong footing, right, in a way. So I think it's also very important to make a right start. And actually for all of you as graduates as well, is or who are graduating and studying, it's important to make the right start, right, in a way, and seek advice of people around you, understand yourself very well, so you know what is the right start to make, right, so you don't waste your next few years as well, right, in terms of what you're doing. And of course, actually, having said that, however, even if you make the wrong start, there is no right time as well to always keep learning, right? I think knowledge and the way things have changed and progressed in the way I had to go from like using Nokia 3210 to like smartphone, right? I mean, all you're good at smartphones, so you have no idea what I'm talking about. But we had like really analog phones in a way. Yes. And before this, I had like before iPhone and Samsung, I had a Palm notebook in a way, which was terrible. So things are adapting and things will keep changing. I'm pretty sure actually when you reach maybe in another 20 or so years, everything is going to be virtualized and digitalized already. I mean, probably you have to learn coding in a way to like do VR stuff or understand in a way how a self-driving car works, right, in a way to make sure it's safe, right? So I think you're always going to need to keep learning in a way, right? So although you may make a wrong start sometimes and you may make mistakes, it's okay in a way, right? Just pick yourself up again. I mean, even if you did a study the wrong course for whatever reason, you can still change course, right, in a way, and keep learning, right? And there's no right or wrong time, whatever age it is, to do a startup, right? Even for the husband and wife team in BioNTech, they started BioNTech when they were 42 years old, right? It's not like they did it. I mean, it's not so glamorized like Facebook in a way where Mark Zuckerberg drop out of college and go and form his own company, right? I mean, most of the time, maybe that's in the fintech world, right? But most of the time, it's not so glamorous in a way, right? Most people, a lot of people even do their startups in their 40s. Nothing wrong with that, right? It's again how you decide and what you want to do, right? Of course, being in a startup is a bit challenging in that sense because you really have to wear multiple hats, right? So personally, for me, I have to wear every single hat in the company, whether it's sales to washing glass tubes to like switching off the lights in a way to like the ULT is alarming in a way, need to go and maintain it, anything you can imagine of, right, in a way. But it's then being flexible and being able to do things, right? Of course, this for me as as a boss perspective, right? But then at the same, for all of you as well, if you decide to join a startup, right? You know, there may be another hat that your boss may put on you and you may feel unhappy as well, right? In a way, like, oh my goodness, this is another thing I need to do. Why am I being able to do this in a way, right? That's a very common thing in even in public sector. How come I cannot able to do this? My boss asked me to do this. Alamak, but it's balancing it and knowing what and how to do in a way, right? So, and it's really a balancing act, right? And in a way, having good time management is being able to do this, and in a way, you will need to do this as well when you become a parent one day, right? Or you need to do it when you do your PhD, right? How do you balance all of the things in a way, right? You need to train interns, your prof wants you to do many things in a way. You need to write your thesis, you need to like talk to suppliers, you need to do every single thing in a way. So, everything is always about balance, right, in a way. And now is a good time in a way to find that balance and know how to balance between different aspects in your life, right? In my last two slides, actually, is there a right time, therefore a right age, as mentioned, to join a startup, right? I don't think so in a way, because no matter what, in a way, like when you said, look at the horizon, follow the rainbow if you want, go and ride it, right, in a sense. And again, the journey for anything and everything is very long. Even look at SpaceX, right? He started in also 2002, and it's only now that he's more commercial success, right? Again, 20-year journey, right, in a way. And of course, he said something which is very, very true. Doing a startup is very like eating glass and staring into a black hole as an abyss, right? It's the most painful thing that you ever have to do, maybe even more painful than having a kid. But it's in a way, it's something that is still, I find very personally satisfying, right, in that sense. So on my last slide, actually, right, just want to share that at the end of the day, actually, no matter where or what you join, it is all a journey, right? And it's kind of like harvesting and having a food, right, in a way. Like in biotech or all of these other things, right? Everything takes such a long time in a way, right? And then in a way, we have to understand what were the factors that came before, who in present in a way is helping to like grow up the tree in a way, who is to come, and who is to really harvest the food, right? But in a way, I would dare say in a way that it is not all of them are equally important, right? Just as you need a good field and good soil in a way to seed, to even allow the seed to seed inside for the small plant to grow into a tree, right? You need good soil, right? Can you say in a way that the person who harvests is more important than the person who like sow and prepare the field? It's really all the same, right? In a way. So it is really a journey in that sense, and it's all hard work in a way. And just because maybe you're coming in the tail end and you do the harvest, then you have to remember that you have reaped the benefits of many people of labor that have gone before you. And the labor is not just within the company itself, right? I'm thankful in a way for all the scientists that have actually helped us in this commercialization journey, right? We have many scientific PIs who are doing research on their own as well in their labs in all different parts of the world using our technology. We have, of course, our internal team in that sense, who are like struggling in a way and doing their best as well, given the circumstances and the situations we are in. We have, clients, collaborators, everybody, they are all laboring together, right? So it's actually one big ecosystem in science, and it's not so insular, right? In a way that, okay, I joined this company, it's only like this, right? Science is very broad and very, very long, just as you see for COVID-19 to commercialize all these vaccines, right? It's an effort of like literally billions of people around the world that are doing this on a daily basis, down from the researcher in the lab, down to the doctor or nurse that injects us, right, in the arm. Everyone plays their part in this whole ecosystem. No one is less or more important than the other, right? We are all actually one big body in this world, right, in that sense. So, that's my concluding remark in that sense, that we are all in this journey together. I'm very happy to meet and speak to all of you all. So we are all in this journey of science together, and we will keep advancing the future of mankind in science. So thanks for your time. I'll be happy to take any questions.
Yeah, thank you so much, Excel. It's very, very inspiring talk, like trying to see how Esco Aster started from a very technologies provider to still technology provider, but in a much variety of sectors. And not only that, they also keep innovating, which is really, really amazing for a startup and continuing to do so. And not only that, they actually also hire NTU graduates, which we are very pleased about. 2 of those are from my lab. So Geraldine is also has been there since the beginning, as Axel mentioned, and then Mridul also. So it's been a pleasure. If you have questions, we can probably take 1 or 2 very quick ones. I'm trying to see if there's anything on the chat box here. Okay, if not, there is only one more question, one more part here, which also is something from Esco, but this is from the Esco venture. We are friends with Esco. So I'm just gonna put up one announcement here that was on request. There will be a pitch competition, and here is available for NTU students.
Yes.
Team from NTU students, you are welcome to submit your startup pitch, and that actually is sponsored by Esco Venture. Yeah. Yeah. Thank you so much. Okay, with that, thank you very much, everyone, for attending this seminar, and I hope it was useful and informative. And if you have any questions or would like to be connected to any of the speakers, please feel free to message me. And I will leave my email here on the chat. Thanks so much. Bye-bye.